A very good afternoon to all of you. Today in the biochemistry part we will discuss about the diabetes millets. You know that what diabetes is. I will give a brief summary of the diabetes. There are two types of diabetes. One is called as type 1 and type 2. Or we can say this is insulin dependent. And another one is non-insulin dependent. In both the cases, either in type 1 and type 2, there is increased blood glucose concentration, what we call as hyperglycemia. But remember one thing, hyperglycemia is not always diabetes. Hyperglycemia can occur due to any condition. But the actual topic here is in type 1 diabetes, there is a problem which is going with this, that is called as ketoacidosis or diabetic ketoacidosis. This is what I am going to explain here, ketoacidosis. How does ketoacidosis occur in type 1 diabetes millets? You know that the normal metabolism of our body goes with metabolism of carbohydrates, lipids, proteins, nucleotides. They all provide us the energy. But the principal component of energy, which provides us the energy, that is the glucose. When there is type 1 diabetes millets, there is deficiency of insulin, absolute or total deficiency of insulin. Due to insulin deficiency, there is surplus of glucose in the bloodstream, but the cells cannot utilize this glucose, either due to deficiency of receptors. We sometimes call it as scarcity in plenty. We have enough glucose available in the bloodstream, but this glucose is not utilized by the cells. Now the obvious problem is cells need energy, they need consistent amount of energy. Brain needs continuous supply of energy, kidney cortex needs continuous supply of energy. So what will be the alternative here? In type 1 diabetes, now the increased fatty acid breakdown, what we call as lipolysis, occurs. Now the fatty acids will give us the energy. Simultaneously, proteins to some extent will give the energy. Now you understand here one point. At the end of lipolysis, what is the end product? That is acetyl-CoA. That is acetyl-CoA. Proteins will also liberate this acetyl-CoA. If you have understood the metabolic part, this acetyl-CoA will go into the Krebs cycle, where the normal process will occur and the energy is liberated. But the Krebs cycle has a limited capacity to utilize this acetyl-CoA. You know that now there is no glucose available to the cells. The only focus is on the lipolysis and protein breakdown. They will liberate a large amount of energy, large amount of acetyl-CoA, which is to be utilized by the Krebs cycle. Simple. But in Krebs cycle, it has a limited capacity. Oxaloacetate is present in limited amounts. Now what will be going here? Some acetyl-CoA will go into the Krebs cycle and that will give us the energy. Now the surplus acetyl-CoA, which is present in the bloodstream now, either in the liver, this will be utilized for the formation of ketone bodies. Now the ketone bodies are generated. Mainly three types of ketone bodies are generated: beta hydroxybutyrate, acetone and acetoacetate. Good. Among the three ketone bodies, only two are utilized by the human body: beta hydroxybutyrate and acetoacetate. Acetone is not utilized by our human body. Now what happens? These ketone bodies also gonna break down. They are high energy compounds. They are water soluble. Once they are formed in the liver, they are transported via bloodstream to the peripheral tissues wherever the energy is needed. Now they will be broken down. Since beta hydroxybutyrate and acetoacetate, they are acidic in character. When they are broken down, they will liberate a number of H positive ions. You have heard about this acid basis. More the concentration of H positive, more the medium becomes, more the blood becomes now acidic. More the OH negative, it becomes alkaline. When a number of these protons are generated, it will make it the acidic. And ultimately, when the pH is changed in the bloodstream, it will lead to acidosis. That is called as diabetic ketoacidosis. I will summarize it in a very short moment. What happens in the diabetic ketoacidosis? There is increased lipolysis breakdown and there is enough generation of acetyl-CoA. That acetyl-CoA is utilized by Krebs cycle, but to a limited extent. The rest of the acetyl-CoA goes for the production of ketone bodies, and those ketone bodies in turn generate more H positive ions and lead to diabetic ketoacidosis. And one thing more I will emphasize here, when there is starvation, ketone bodies are also generated at that point of time, but starvation will never lead to diabetic ketoacidosis. They can never go for the ketoacidosis. Thank you so much.